Cancer Genetics, Part 1, Module 3, Lecture 1 in the Genetic Series. So cancer prevalence, it's estimated that 1 out of every 3 individuals will develop cancer at some point in their lifetime. The risk of cancer varies from different states. There's probably environmental as well as genetic factors for the reason for this. Here's just an estimate of the leading causes of cancer. In males, it's prostate cancer. In females, it's breast cancer. The second for both is lung and bronchus, followed by colon and rectal, urinary in males, and uterine in females. As far as death, lung and bronchus has a higher death rate, as with prostate and breast being second, but those are cancers that we're better able to treat, and therefore, even though they're more common to occur, the actual death of lung and bronchus is higher in both males and females. So cancer definitions: neoplasia, disease process characterized by uncontrolled cell proliferation leading to a mass or tumor. Uncontrolled unregulation of cell growth results in a mass. Not all neoplasias are cancerous. The word malignant is defined as uncontrolled growth that's capable of invading other surrounding tissues and traveling to other sites. So that's the difference really between benign and malignant. Is benign growths just keep growing, but they don't necessarily invade other tissues or go to other places. Travel to other sites is often through the lymph nodes. Metastasis is defined as neoplasia that spreads to distant sites. For instance, breast cancer then metastasizes to the liver. A benign tumor is a tumor that does not invade or metastasize, but it still can cause problems, especially if it's in confined space. For instance, a benign tumor in the brain, if it keeps growing, can push on other structures, and because there's nowhere to go, it causes a lot of problems. So types of malignancy: sarcoma is a form of mesenchymal tissue, like bone, muscle, connective tissue, nervous system. Carcinoma, epithelial cells that line organs such as breast and intestines. And then hematopoietic and lymphoid malignancies spread through the blood and bone marrow, such as lymphoma and leukemia. So let's talk a little bit about the cell cycle. So we start the cell cycle with cell division or mitosis. Then we divide into 2 separate cells, which is cytokinesis. G1 is one of the most important times in the cell. This is when, after we divide our cells, it's like the double, we make sure that everything is correct. We then replicate our DNA, and then this is double checking that we didn't make a mistake. Cancer cells sometimes try to bypass some of these checks, and this leads to uncontrolled proliferation. This can occur in a number of different ways. So, oncogenesis. So we have heredity. There are other factors that can lead to proliferation and growth versus cell death by apoptosis. Chemicals, tobacco, radiation, UV, viruses can all affect this balance between proliferation and cell death or apoptosis. And it's this balance that's critical. All cancer is genetic, but not all cancer is hereditary. The actual cancer cells usually have a change in them that allows them to never stop growing. So a normal cell grows, it divides about 10 to 20 times and then stops growing. Cancer cells, they don't follow rules. They like to divide forever. They like to spread to different places. So here we have a normal cell. And if we have a change in the gene, we're probably still okay. But it's when we have a second change and sometimes a third change that we have the cancerous cells. So if we actually looked at the genes in a cancer cell, there are often many changes: loss of chromosomes, deletions, amplification of certain genes. But the difference between inherited is that you're born with some of these predispositions. suppressor genes. So tumor suppressor genes are important genes to kind of stop cancer from growing. And many of the cancer predisposition syndromes are because of loss or malfunction of these tumor suppressor genes. Both alleles or both copies from each parent need to be abnormal before you exhibit the uncontrolled cell growth. So in hereditary cancer, loss of function of the second allele is usually a somatic event that occurs later. Most hereditary cancers result in mutations in these tumor suppressor genes that result when, so we're born with one copy that doesn't work, but then in the cell, if either by bad luck or radiation or whatever, we lose the second copy, that's when we have loss of controlled growth. And this is the Knudsen two-hit hypothesis. So it takes multiple hits before the abnormal growth occurs. So in normal cells, and this is just everyone, breast cancer, for instance, 1 in 9 women will develop breast cancer. We're going to talk about that a little bit more later. But most of those 1 in 9 women are born with 2 normal genes for tumor suppressors. But then over the course of life, bad luck, 1 is damaged, but we're still okay. But then in a small group of cells, especially if that small group of cells is in the breast, we lose a second, and then the tumor develops. In hereditary cancer, you already start out with 1 knock against you. You start out with 1 copy of your gene that doesn't work correctly. And that only takes 1 event to get into trouble. And that's why, even though, for instance, in the case of breast cancer, 1 in 9 women develop breast cancer, if you're born with a BRCA1 gene, which is a tumor suppressor gene, your risk of developing breast cancer is far greater. So oncogenes, proto-oncogenes normally promote growth and the survival of the cell. These are activating mutations. Proto-oncogene is inappropriately activated, resulting in, again, we talked about that balance, the balance between growth of cells and apoptosis. So it stimulates proliferation and prevents apoptosis. Here is a diagram. The vast majority of cancer is sporadic, which means just something bad happened. About 10 to 20% are familial, which means there's probably some genes, but we're not really sure what they are. And then this small sliver is the hereditary cancers, the BRCA genes and so on. So, sporadic versus familial cancer. Sporadic results from a buildup or damage of cells over time. Typically, the cancer is in older individuals, usually diagnosed with the cancer after age 50. They're common cancers, breast cancers in women, prostate cancer, for instance, in men, and not a lot of family history of cancer in the family. Familial is a combination of genetic, environmental, and lifestyle factors. Not a single gene is inherited, and typically it's sporadic, but there's more cancers in the family than we would expect by random chance. And finally, we get to hereditary cancer. This results from an inherited, most of the time, gene mutation that predisposes a person to develop cancer. Some hereditary cancer syndromes are a result of a de novo or new mutation. Hereditary cancer syndromes have reduced penetrance. The mutation does not skip generations, however the cancer may. Because, as I said, there takes 2 events to occur to get the cancer. So, if somebody carried a gene, for instance, the BRCA1, had a child when they were, 22, and died from a car accident at 25, they may have never had cancer, but they could still pass it on. 100 different genes are known to be associated with this increased risk of cancer, and this is an ever-growing number. Usually when I talk to families, I tell them today is, this is what we know today, but I don't know what we're going to know tomorrow. So hereditary cancer's clues for personal and family history. Things that would make us and a patient be more concerned that this may be an inherited cancer. So family history that would make us more concerned that this is an inherited cancer include a young age of diagnosis, for instance, the individuals under 50. Multiple blood relatives with the same type of cancer, either all on the maternal side or all on the paternal side. Close blood relatives, first, second, third degree relatives. Consider other risk factors as well. For instance, multiple lung cancers in a family that smokes is not as concerning as lung cancers in individuals that do not. Individuals with multiple primary cancers, related cancers in the family, for instance, breast and ovarian, and rare cancers such as male breast cancer. So, what is the most common cancer? Familial, teratogenic, sporadic, single gene, or multi-gene? This concludes Lecture 1 in Module 1.